Hi guys, so just like I told you in the last video, I told you we should solve our problem in this video. Well, don't get tired yet. I'm sure these guys just been talking about method of science, data collection, everything. But I said it, you have to be meticulous as a scientist, so don't get bored. Biology is actually very, very interesting. Well, I love reading biology, so the fact that I discover new things about organisms around me, even about myself, the body which my body, so I love discovering that. So now we have done experimentation. Now we have tested out the hypothesis. So I believe we did good job. So our hypothesis was correct, yes. And if yours was not, do it again. Just go over that again. So the hypothesis was correct. Now let's go. We have done experiments. We have got obtained results from the experimentation. Now the next thing is that the next stage is the analysis of data. So what you have gotten, it's just like in the data collection stage when you were just collecting raw data, you still have to organize it. Now I'll say the data now is information now because it has been tested out, so it's more like an information. We can work with it. That's it. We can work with it. Now when I mean work with it, we can apply it now. You understand? So now we're in the analysis of the data. Now analysis of data is also very important because it tells you can then look at which information you collected was reliable or was not reliable. You understand? So that is the importance of analysis of data. It also tells you where you need to do further research on, or areas where research is quite okay. So you can work with that. Or this area we still need to look further into this. It will tell you that okay, this information is not really reliable. We don't really need this. Okay, we need this information. So that is more like what you're doing the analysis of data. Now, biology is a multidisciplinary science. I told you, you need knowledge of chemistry, you need knowledge of maths, you need knowledge of physics. In this same phase or stage of scientific method, you also be needing knowledge of statistics. Statistics is purely math, so you'll be needing knowledge of statistics. You'll be looking at quantiles, you'll be looking at mean deviation, you'll be looking at averages, mean, and the rest, because you want that information which you are presenting out to be meaningful. Because one thing you should know is that, like when you write academic papers, you write research papers, there's something which they'll tell you that a good research paper is one in which if a layman should pick it up, he will understand what is actually going on. So, and that's really funny because you expect that you should pick up scientific papers and they should be so bogus on the rest. But truly, you should do it in a way that the layman that picks it up understand it, and you can only do that if you properly analyze the data. Do you understand? You can only do that if you properly analyze the data. And of course, you have tools like graphs, you have tools like charts and tables which can also help you to probably present a good analysis of the data. And finally, in the method of science, you make an inference. Now, inference is just a conclusion of scientific facts. Now, there's a difference between inference and hypothesis. Now, I mentioned earlier the other time that hypothesis is a statement that explains an observation and it also describes ways in which it can be tested. That means an hypothesis can be directly tested. That's where it's different from an inference. An inference cannot be directly tested. So you test an hypothesis, you don't test an inference. So an inference is more like a conclusion of the scientific fact. Or we have done thorough research, or we have done the data collection, we have been able to make hypotheses, we have been able to experiment, we have analyzed, and finally we have concluded that this disease outbreak is caused by this particular virus. It is transmitted through the air or respiratory droplets. Okay, it is potent in the sense that the mortality rate is high and the survival rate is low, and it is a disease outbreak. That is, it is something which is a major problem and should quickly be addressed. This is our conclusion. That's an inference, so you can't directly test that. We have concluded it already. Do you understand? It's different from hypothesis that is saying we believe that the reason why people are coming down with this disease is because of poor environment. We believe that what is causing this disease should be a bacterial particle, should be a bacteria. That's what we believe. Now, you believe that it's more like an hypothesis. You are believing that this is the reason why. So you need to test it out. But at the stage of inference, you have tested it out. So you are sure, you are very, very sure that, oh, this disease is caused by bacteria. This disease is caused by a virus. This disease is transmitted airborne. This disease is potent. The mortality rate for this disease is high. The survival rate is low. It's an outbreak and it should be addressed immediately. You are very sure. You are so confident because you have done the needful and you know this is the conclusion of what you have done. And also in inference, you also add, you suggest solutions. During the inference, you are suggesting solutions to the problem. So we need a vaccine that will eradicate it so that the antibodies can effectively fight against this infection. We need to carry out safety precautions so as to prevent the further spread of this disease. So you are at that point in time, you are suggesting, you are making suggestions that can effectively solve the problem. So we have solved the problem. The problem was there, we have solved it. And being that we're meticulous in whatever we're doing, we ensure we didn't create problems at different stages. Particularly at stages which are critical, like data collection stage, there's no problem. We organize the data. From the stage of the hypothesis, we made hypotheses, we tested them out. If they were not effective, if they were ineffective, we either refined them, revised them, or we discarded them. We carried out experimentation under controlled environment. Now that's very important. In science, we don't just do experiments just like that. Yes, we are testing out hypotheses, but these experiments are always done in a controlled environment. That is, there's something we call the control experiment. Just like when I said, if you say that the experience should be given subcutaneously or rather intravenously to produce the greatest response, that means we will not just give them intravenously and probably we just say, yes, yeah, you could see that they responded. How will I know? So what we do is that we do it under controlled environment. We have some certain populations of people, we give them orally. We have another population of people, we give them subcutaneously, that is under the skin. So those two populations, we can call them the control experiment. So that means if we are going to be making a statement, we are very sure because we have effectively tested that out. So just as I stated, experiments in science is done under a controlled environment. Also remember, in science, we do careful observation. So those two things, please don't forget, they are very critical. Now, so that's all on method of science. So I'll go over them again. The first method is the first stage in the method of science, or the scientific method, the investigation stage. Number 2 is the data collection stage, and these has 4 aspects: observation, measuring, sampling, and organization of data. The next stage is hypothesizing, that's making hypotheses, statements, explaining your observations, as well as describing how they can be tested. Next stage is carrying out experiments under a controlled environment. Do 2 major things: to test out the process of the hypothesis, to test out the prediction of the hypothesis. Next is the analysis of data. This is where we determine which information is reliable or not and where we need further tests. And also, during the analysis, you also compare what you get from what researches which were done previously. And finally, you make an inference. It's more like a concluding statement of facts. And also, under your inference, you also make suggestions on how this problem can be solved. So, don't forget, each stage are very critical. I'll see you in the next video.